Hello everyone. So in this video, what we're going to do is spend a little time walking through the FRQs that will appear on, or that may appear on the Unit 5 test. So one of these 5 FRQs will appear on the test. So just make sure you're prepared and ready to write for each of them. So looking at FRQ1, it reads, in corn, a gene for yellow kernels is dominant over the gene for colorless kernels, and a gene for smooth kernels is dominant over one for wrinkled kernels. 1A, describe a controlled genetic experiment to demonstrate that those genes are linked. Now, they're not necessarily looking for you to come up with your own experiment on this. If you want to have an experiment that shows that 2 genes are linked, an awesome example is on page 301 of your textbook. Now, they're asking you about corn, but in reality, this is a Hunt Morgan fruit fly question, okay? So there it is. I would absolutely pattern this after the experiment outlined on page 301 in the book. It's figure 15.9. So please use your textbook and reference that figure on page 301. So it says, describe the genotype and phenotypes of the P generation. Well, again, what do we see here in the P generation of flies? One parent is homozygous dominant, the other is homozygous recessive. So you can apply that to the P stuff, or not the P stuff, the corn. Sorry, getting my wrong starchy vegetables set up here. 2, describe the predicted genotypes and phenotypes of the F1 generation. Okay, well, if the parents are all homozygous dominant or homozygous recessive, what would you expect in your F1 offspring? Roman numeral 3, describe the predicted phenotypic ratios for the F2 generation. Oh, in the blue box here, predicted ratios of test cross offspring. So they've got it for you right there. All right. So again, that really makes a huge difference for you as you go and prepare this. And again, prepare yourself. Okay, make sure you're ready. B, genetic evidence indicates that these genes are linked and that their recombination frequency is approximately 4%. What does it mean for genes to be linked? So make sure you explain that. C, describe the process of crossing over and explain how the recombination frequency is determined. So how did you calculate that? We did that in the chapter 15 end of chapter questions. So what do you do to calculate recombination frequency? And again, what does it mean for crossing over to happen? Sort of explain that. All right, let's see. Number 2, describe in detail the process of meiosis as it occurs in an organism with a diploid chromosome number of 4, 2n equaling 4. Hey, when you guys did the drawing on the lab tables, that's what you did, right? You set up a drawing with a diploid number of chromosomes being 4. So include labeled diagrams in your discussion. So you do a rough diagram and make sure you label it. You get a couple points for that. Indicate when and how each of these events occurs. So crossing over. When does crossing over happen? What's going on? Independent assortment. When does an assortment happen? What's going on there? Segregation. When you separate those homologous chromosomes. What's going on there? When does that happen? And then with non-disjunction. When does non-disjunction occur? And what's happening with non-disjunction? So this is going to be a chapter 13 question, right? So if we look back at chapter 13, pages that'll be really helpful for this, 260, 261 are golden. They're really good. And then 263, the figure there, I think can be useful as well. So just make sure you're going over when and what's going on in each of those scenarios, and you have briefly labeled or quick diagram with some labeling. Number 3, assume that a particular genetic condition in a mammalian species causes an inability to digest starch. Okay. This disorder occurs with equal frequency in males and females. Boom, boom, boom. That's a clue. In most cases, neither parent of affected offspring has the condition. Another clue. Okay. A, describe the most probable pattern of inheritance. So if it's affecting males and females equally, what does that suggest? If parents don't have it, but their kids do, what does that suggest? And make sure to explain why. Okay. It says include sample crosses to sufficiently verify your proposed pattern. So what they want you to do is say, okay, if this is the pattern of inheritance and you have this type of cross, these would be the types of results that you would expect. So you can have like theoretical parents, theoretical Punnett squares, and then show what you get, and then explain why. Make sure you explain why in words. And then B, explain how mutation could cause this inability to digest starch. What's doing the work in cells? Enzymes. Enzymes are proteins, and proteins get their instructions from DNA. So talk about how a mutation in the DNA could impact, proteins and the ability to digest starch. All right, number 4. Here's an FRQ near and dear to my heart. This is the FRQ from the AP Bio exam from the year I graduated from high school. So you can see how advanced graphics were in problems back then. All right, so it says it's an organism heterozygous at 2 loci on different chromosomes. So it's 2 genes on different chromosomes, and the individual is heterozygous. So I've got a dominant A from one parent, recessive A from another. On a different set of chromosomes, got a dominant B from one parent, recessive B from the other. A, explain how these alleles are transmitted by the process of mitosis to daughter cells. So again, maybe it was 261. No, I'm sorry, 263. 263 might be a nice reference page because it sort of reviews what's going on with meiosis. Look at how the chromosomes are lined up. Okay, that's a big thing there. Part B, explain how these alleles are distributed by the process of meiosis. Again, 263, look at how the chromosomes pair up. C, explain how the behavior of these 2 pairs of homologous chromosomes during meiosis provides the physical basis for Mendel's 2 laws of inheritance. Mendel's 2 laws of inheritance, one is segregation. An organism may have 2 copies of a gene, but only passes on 1 copy at a time, because think about what happens during meiosis 1 to homologous chromosomes. Sort of explain that. The other law that Mendel came up with is his law of independent assortment. How 1 pair of homologous chromosomes lines up has no effect on how another pair of homologous chromosomes lines up. And again, think about the why. Okay, so that's a good one. They're all good. Number 5, discuss the following phenomena in which sex chromosomes are involved with particular reference to their significance or consequences in humans. So we're dealing with sex chromosomes in humans. A, how does this play a role in sex determination? So, what do males always pass on? What do females always pass on? What determines whether a person's male or female? And, why do you have roughly equal numbers of males and females? Stuff like that. B, sex-linked inheritance. Again, you want to discuss how genes may be found on the X chromosome, and since males and females have different numbers, they're affected differently, and why. So get into that kind of stuff. With that, I might talk about why males are more likely to have sex-linked recessive conditions and why females are more likely to have sex-linked dominant conditions. C, formation of Barr bodies. This is something that was discussed in the lecture videos, but the idea is a female may inherit 2 X chromosomes, but one of them gets randomly shut down permanently. So when that happens, you have 2 X chromosomes, but you shut down one. And so, what's the impact of that? You can look at the idea of chimerism with certain conditions. So that's something that's interesting. Why you don't see Barr bodies in males typically, stuff like that. D, variation in the kinds and numbers of sex chromosomes. So with that, that's looking at aneuploidies, like having 1 X chromosome, which is Turner syndrome, having multiple X syndrome, not syndrome, but having females that are like XXX, and what goes on with that. What would you see under the microscope? Males, how you can have males that are XXY with Klinefelter syndrome, or males that are XYY or XYYY. And the relative impact on that. So just some interesting possibilities there. And finally, E, explain the difference between sex chromosomes and autosomes. So, what's the difference in the complement that individuals receive? What are differences in patterns of inheritance? Stuff like that. So lots of ideas to explore there. So lots of points that you can get. All right, as you work through this stuff, if you have any questions, let me know. Otherwise, hope you have a great day. Thank you.